Hello friends, this is Richa here and you're watching Learners Are. Here I'm right back with my third part of our topic, discovery of the genetic material, which consists of Hershey and Chase experiment. So our today's topic is Hershey and Chase experiment. So before starting our topic, wishing you a very happy and a safe Dashera guys. So let's start our experiment part. Alfred Hershey and Martha Chase conducted this experiment in the year 1951 and 1952 to know the nature of the genetic material. So, first let me tell you what was the need for this experiment, why was this experiment carried out. Although it was proved with the Avery's experiment that DNA was the transforming principle which transformed the R cells into the S cells. But there were some scientists who were not convinced that particular with that experiment and didn't accept it that DNA was the transformation principle. Therefore, this experiment was carried. So let's begin with the experiment part. For this experiment, bacteriophage viruses were used. Before using them for the experiment, they were radioactive labeled. Now, first, let me mention that what is radioactive labeling. When we radioactive label something, it means that the particular substance which is radioactive labeled will mark the presence of that particular substance. One of the virus was radioactive labeled with S35 isotope of sulfur, which indirectly labeled the protein of the virus as the sulfur is present in the amino acids. Similarly, the second one was radioactive labeled with P32 isotope of phosphorus. As we know that the phosphorus is present basically in the DNA as it forms the backbone of the DNA. So it radioactive labeled the DNA of the bacteriophage virus. Now we'll come to the experiment part. Now this experiment consists of 3 steps. First step is the infection. Guys, infection is the process during which the virus infects the bacterial cell and transfers its genetic material to the host cell. There may be some students who might not be knowing about this infection process, and if you know, it's well and good, but if you don't know, don't worry. You don't panic. You will be studying about it in higher classes, mostly graduation or in degree. So as it is a complete topic in itself, so if possible I will also create video lectures on this particular topic which is related to the life cycle of the bacteriophage viruses. It is a complete different vast topic, so I'll make it in a next video. So it's something secondary guys. So for now let's focus on our current topic. So after infection, the next step is blending. Blending is done to remove the viral particles from the bacterial cell, as we only want the bacterial cell and not the bacteriophage viruses for the experiment. Then comes the third part of the experiment, that is the centrifugation. It is the process where where any liquid content is rotated at high RPM at a supernatant, which is the lightweight content, and a heavyweight content, that is the pellet, is obtained. The supernatant basically contains the viral particles whereas the pellet contains the cell debris and the other cellular components like the viral DNA, host DNA, and other components of the host. Now the experiment is done, so let's come to the observation and the conclusion part. So after centrifugation, the scientists observed that in one of the test tubes in which S32 radioactive labeled virus was present, the radioactivity was shown in the supernatant, whereas in the second test tube, which was radioactive labeled with P32, the radioactivity was shown in the pellet. So, as I already mentioned earlier, that the radio labeling bacteriophage with P32 isotope, indirectly radioactive, labeled its DNA, and also the pellet contained the DNA and the cellular components. Also, the radioactivity was shown in the pellet. So all these points indicates that the DNA was the genetic material which entered the bacterial cell, rather the protein which not yet entered the cell debris and remained in the supernatant form only. Hence, with this experiment, it was proved experimentally that DNA is the genetic material. So friends, this was the end for our discussion regarding this topic, that is discovery of the genetic material. But before signing up guys, I would like to mention one thing, that here I have uploaded the link for my scanned copy of my notes, also for the previous parts of this topic like Griffith's experiment and the Avery experiment too. So you can refer them. You can get these from the description box below the video, so you can refer those notes. But it's my suggestion guys, if you create your own notes, because if you'll just copy them, then it will not be too useful for you. Those notes are very concise in content guys, so and they're very to the point. So I suggest that if you refer those notes, try to create your own notes, refer them, grab some more information if you want to from other resources like books or internet, and also try to understand them more clearly. Try to manipulate them in your own words and create your own notes, taking help from those of my ones, of course. So meet you soon in the next video guys with another new topic, and don't forget to like, subscribe and share. Till then, bye.